Welcome back, everyone from coffee. I'd like to welcome all of you to Thematic Session 4, which is on protein and tissue engineering. Our first speaker is Paul Carter from Genentech.
Let me begin by thanking Jim Wells and the other organizers for the opportunity to speak at this fabulous symposium. For more than 30 years now, I've been working in developing antibody therapeutics, including bispecifics. And I feel very blessed at being working in the field at a time where, as a field, it's made such astonishing progress. And there are now more than 100 antibodies that are approved as therapeutics. So you might be wondering, well, is there anything important left to solve? And I guess really the main message for my talk is, yes, there is. And some perspective on kind of the broader fields kind of offer in this review article in Cell with my colleague Avindra Rajpal from back in July. What I'm going to do today is to talk about one specific problem in the field, and that relates to high concentration properties. And specifically, I'm going to talk about a bispecific antibody, and the particular property we're going to focus on is viscosity. Before I get into this sort of vignette, I need to give you a brief introduction why viscosity is a major consideration in subcutaneous delivery of antibodies, including bispecifics, and as to that, a little background on bispecifics. So you look across the antibodies which are currently approved, roughly 2/3 of them are given by intravenously. And almost all of the rest are given by subcutaneous delivery. And this fraction is actually growing, and it's not hard to understand why. For subcutaneous delivery, it can be done much more quickly, in a few minutes, compared to a few hours typically for IV. It's also possible to self-administer at home, in some cases using pre-filled medical devices. And obviously, this is much more convenient for patients and their caregivers. However, subcutaneous delivery comes with some pretty significant technical challenges. Firstly, you're limited to small dosing volumes and fine-gauge needles, and sometimes ultra-high concentrations so that you can deliver the dose required. So even as high as 200 mg per ml. And this can lead to high viscosity and delivery back pressure with injection site pain. So an inconvenient truth is that many antibodies are not well behaved at these ultra-high concentrations, and they can lead to high viscosity, aggregation, precipitation, gelation, and opalescence. So a little background on viscosity itself. It's probably a topic that may be not that familiar to many of you. It's commonly measured using a rotary rheometer. It's the ratio of shear stress and shear rate. The units of measurement are typically centipoi. And viscosity is impacted by a number of different parameters, and I'll talk about a few of these today. In thinking about viscosity, there are a couple of working limits. One of them is in manufacturing, which is roughly 20 centipoi, and for subcutaneous injection, it's lower, roughly 12 centipoi. And to give you some idea of what these numbers actually mean, you can kind of relate them to everyday liquids. And I think you probably get an appreciation that these limits are really not very high. Brief background on bispecifics. There are literally 300 or more bispecifics in clinical development, and actually even 30 or so trispecifics or multi-specifics. Of these, there are at least 7 bispecifics which are approved. So this is a time so bispecifics have been around really since about 1960 as a concept, and it's really only over the last few years that they have started to come of age. There are an astonishing number of formats which kind of reflects the ingenuity of lots of different groups around the world. There are literally more than 100 different formats which have been published. Certainly a much smaller number of these have actually made it into clinical trials. Of these, the bispecific IgGs, which I'm illustrating in this cartoon, have certainly been widely used, and it's a format that we've widely used. And so this is our 4 chains, so it's a hetero tetramer. So the short vignette, which I'll share over the next handful of slides, relates to a bispecific anti-IL-13, anti-IL-17. And here we're looking at the viscosity. Here are the parent antibodies, about 8 centipoi. You can see that the bispecific at the same concentration is roughly twice the viscosity one would expect, and that's actually also true of a mixture of the 2 parent antibodies. And so kind of set out to try to understand why this might be, and then to try to figure out how to overcome it. A little bit of background on the kinds of non-covalent interactions that contribute to viscosity. Thanks to the efforts of many groups, really going back over their 30 years, it's now fairly well understood the kinds of forces that can be involved include salt bridges, hydrogen bonds, hydrophobic effects, and I think to some extent from our own work, also that cation pi and pi interactions can also contribute. So how do you overcome high viscosity? And well, there are at least 4 different approaches that can be applied for antibodies. I'm going to talk about 2 of those today, highlighted in blue. So you can add excipients, and we'll focus on salt and hydrogen hydrochloride to attenuate the non-covalent interactions. And then you can also engineer the antibody to reduce viscosity, and there are a couple of other approaches which I don't have time to cover today. So first, looking at excipients, and let's focus on this side. So this is their the bispecific. This is their parent in grain buffer alone. If you add sodium chloride, it does reduce the viscosity a little bit, but it's much more pronounced in the presence of arginine hydrochloride. And the fact that the NaCl and arginine hydrochloride behave rather differently suggests that it's more than just salt bridges. So one of the next things we did was some molecular dynamics and looking at arginine interaction with the Fab fragments of the IL-13 and IL-17, and then basically asked from these molecular dynamics, where does the arginine spend its time? And you can see that there are some sort of hot spots with some aromatics and also some other, not just aromatics, but polar residues in the VL of IL-13. And of course, we looked at the IL-17 antibody as well. We also considered the structures that are available for the antibodies bound to their ligands, and then designed a series of mutants to kind of explore interactions that might impact viscosity. In the interest of time, I'm just going to talk about a few variants, specifically of some aromatic residues, which, when mutated individually, actually very significantly reduce the viscosity down to roughly that of the parents. Some of these are in the antigen binding complementarity determining regions and interfere with binding, but there's at least one here which doesn't. So let me wrap up by saying that what I've tried to do today is give you a sort of a vignette of a project which is looking at a high concentration property, which is of kind of growing interest in the antibody field and arguably underexplored. So we have an example where it's anomalously high viscosity. We did some molecular dynamics simulation. We found some point mutants which reduced viscosity, and some of those also preserved antigen binding. We've also extended this to other antibodies, and in more recent work have done a much more comprehensive survey of this antibody, which is still ongoing and a little too early to comment on. And we hope that this method will complement some of the other methods to reduce viscosity. And it just remains for me to thank the people who did the work specifically. Line share was done by a former postdoc, Schepartz, Novo. And with that, I'll stop and thank you.
Thank you, Paul. Our next speaker is not Matt Francis. It's my colleague, Evan Miller from UC Berkeley.
Wonderful. Thank you for the introduction, and thanks to the organizers for the invitation and the chance to be here. And thank you all for being here. My name is Evan Miller. Title of my talk is Fiat Lux, Fluorescent Indicators to Watch Cellular Physiology. And I have to admit, it's wonderful to be here with you all, and I feel a little bit rusty. I haven't done this in front of, actual audience in a while, so please bear with me. First, I want to thank my group. Here's a picture of us. Never trust your PI, especially when he suggests going to do ocean kayaking. It's very different than flatwater kayaking. But I'm really fortunate to work with a great group of students and postdocs, and it's been a difficult 3 years, and I just appreciate both the science that you've done and the way in which you've taken care of each other, so it's been really fun to watch. And with that, I'll tell you a little bit about what we do. So we're really interested in using light to spy on cells, to watch biochemical and biophysical processes in action. And so we think about how we can build dye molecules that can read out biochemistry and biophysics in real time. And one of the things that we're fascinated by is this idea of membrane potential. And it's really central to life. All cells spend a significant portion of their ATP budget on pumping sodium and potassium ions in different directions to set up this negative potential inside cells. And this is tough to estimate, but somewhere between 10 and 50% of cellular energy budgets go toward maintaining this membrane potential. And so we're fascinated by being able to study this with higher precision. And so this is important, of course, in a couple different areas that you might readily recognize, like the brain, or in muscles like the heart. But it's also, as I mentioned, really important for non-electrically excitable cells, or all the other cells in our body as well, where a substantial portion of our energy is going toward maintaining this membrane potential. So if you want to study membrane potential, this is usually done with an electrode, and you impale a cell with a pipette that's got some sort of internal solution, and it's hooked up to an amplifier, and that gives you really perfect electrical access to a single cell. And this is perfect for single-celled brains, but it's really invasive and pretty low throughput. It's especially difficult for contexts where you might want to look at many cells at the same time, or where you're interested in areas that are not at the soma, maybe some sort of distal area, cells that are moving, or any sort of intracellular membrane, which is something that we'll talk about in just a minute. So this is the technique in action. Here's a picture of our microscope and our amplifier, our head stage, a micromanipulator that allows us to bring that pipette in close contact with a single cell. If you want to do something that's a little bit more complex, then you have to do some engineering. This is not in my lab. And it's really easy to scale this, of course. You can get to 12 neurons or 12 cells with just a little extra work. So it's really difficult to do multi-cell recording, and so we're interested in sort of developing complementary approaches to doing electrophysiology where we can do voltage imaging directly to take a picture of or look for changes in membrane potential. And increasingly, we're thinking about how we can actually measure values of membrane potential. And so we take a chemical approach to doing this, where we build fluorescent dye molecules that intercalate in plasma membranes. And the idea for this was described by Liang-Shi Li in 2007, where he had this really beautiful paper describing mesoscopic electron transfer as a method for looking at membrane potential. And so the idea is that we build a fluorophore that sits in the plasma membrane. There's a potential that exists across the cell where we have negative on the inside, positive on the outside. We can excite that fluorophore, and an electron will move from one side of the molecule to the other to quench or short circuit that fluorescence. In this case, the electron moves quickly because it's aligned with that plasma membrane potential. And then when a neuron depolarizes or a muscle cell depolarizes to evoke a contraction, now that membrane potential is changed, and so now the voltage opposes the movement of the electron, and so it takes longer for that electron to move, which gives the fluorophore a chance to emit a photon. And so our hypothesis is that changes in membrane voltage are going to alter this rate of electron transfer, or PET, and that modulates the fluorescence. And so we can look at fluorescence. It tells us something about the underlying rate of electron transfer and tells us something about the change in the membrane potential. And so these are the types of molecules we make. Gives us a turn-on response to those depolarizations. It can be very fast because it has to go during the nanosecond lifetime of a fluorophore. We can use any color we like. We're moving very quickly, so we have very little capacitance load, and we can be quite sensitive if we tune those electronics. And so again, here's one of the voltage-sensitive fluorophores we make. Wide field microscopy image of a hex cell. We look for this chicken wire pattern of staining. It tells us that the dye molecule is in the plasma membrane. That's where we want it to be, to look for changes in membrane potential. And then we stick an electrode on one of these cells, change the membrane potential, and ask how that fluorescence evolves with time. And what hopefully you can see here is that at depolarizing potentials, so more positive, it becomes brighter. At hyperpolarizing potentials, it becomes dimmer. It's roughly linear, nice even stair steps at those different membrane potentials that we're examining. And so we're interested sort of in 4 areas to give you a broad overview of the lab, thinking about sort of chemistry and understanding how these fluorophores are working, how we can apply this in the context of the brain, in the context of excitable cells like the heart, and also in non-electrically excitable cells. So I'll tell you just a little bit about some of the chemistry. We like to work with different colors, and so we've worked hard to develop a palette of colors so we can plug and play with a lot of the existing tools that are already sort of widely used, especially those that work well here. We can offer the ability to look in sort of a different channel, a different color. More recently, we've been thinking about some gaps in the literature, and so carboxyfluorocene, which many of us have used probably, was first synthesized in the 19th century. Sulfonofluorescein shortly thereafter. We were interested in this other biologically relevant acid, phosphoric acid or phosphonic acids. It turns out it hadn't been made, so Josh Turnbull, a recent graduate in the lab, figured out how to make this. We really like this molecule. It's much more water soluble. It doesn't spirocyclize, which is important for us to put things on plasma membranes. We have an additional sort of functionalizable handle with the 2 acidic sites on the phosphonate. And we get really nice retention inside living cells, so comparing the fluorescence of AM esters of the carboxyfluorescein and the phosphonofluorescein, we get really nice retention. And so Josh is working, and we have a ChemArchive paper describing rhodamines that we've done with the 3-phosphonate group. So now I want to tell you a little bit about what was initially a disappointment, but turned into something that we think is really interesting. And so as I mentioned, we're interested in looking for this chicken wire pattern of staining. We want it to be on the cell surface so we can measure plasma membrane potential. And one of the first projects that we worked on in the lab was to make these rhodamine type dyes. This is work that Parker Deal did. And he worked really hard to get these dyes onto the plasma membrane. But actually some of the first ones we made were all internal. And so this was really disappointing because we thought, oh, we'll just put on a rhodamine. We have our molecular wire. We'll start to do some voltage sensing in other colors, but everything was inside. Again, here with these esters, everything was localizing to intracellular membranes, and this was totally useless to us because these were all broken. They weren't where they were supposed to be. However, we eventually realized that we could take advantage of this and sort of repurpose and redirect these guys to be able to look at changes in membrane potential in internal organelles. And so we wanted to redirect these rhodamine voltage reporters. And this is work that Paul Vaclayer did to redirect these to mitochondria. And what I'll tell you about in our time remaining is his efforts to target endoplasmic reticulum. And so this is a collaboration with Alanna's group, thinking about how we can use some of their lipid labeling methods to target internal organelles. And so Paul's idea was to take this rover tetrazine. This electron-rich tetrazine provides another source of quenching, and so it makes that fluorophore very dim. And then we could use a transcyclooctene appended to a targeting group, which I'll talk about in just a minute. We can do the sort of click reaction, the inverse electron demand Diels-Alder reaction to generate this more electron-poor heterocycle that no longer quenches the fluorophore. And so we get a 6-fold turn-on and an increase in brightness. And that's what I'm showing you here. The absorbance doesn't change, but the emission properties do change. And so when we add that TCO, it becomes brighter. And so what we could do is we could preload cells with ceramide TCO. Alanna's group had shown that this will stain the endoplasmic reticulum, especially above 37 degrees. And so we could pre-incubate with TCO and then add our rover tetrazine. We get uncaging and ligation only in the ER. And so Pavel named this ligation unquenching for activation and redistribution, or lunar rover. And Pavel's now fittingly a postdoc at NASA. All right, so Lunar Rover localizes to the ER. So here's Lunar Rover overlaid with burst, which goes to plasma membranes. If we overlay Lunar with ER green, we see nice white pixels there indicating reasonably good overlap. Here's some quantification showing high correlation between Lunar and ER green, and lower for the plasma membrane potential, about half of that, and close to 0 for that 90-degree control. So we did some calibrations that I won't tell you about now, but one really surprising experiment was when we went to go patch these cells. And so this is work that was done by Annalise Guest in the lab. And so a patch clamp hex cell, we're changing that plasma membrane potential, and we were actually sort of surprised to see that Lunar Rover changes its membrane potential. And so what you'll notice is it's going down at depolarized potentials. So this was opposite of what we typically get. We did a control experiment where we used a Lunar Rover 0, and so this doesn't have the electron-rich aniline, that electron source that makes it voltage sensitive. When we run this experiment, it's a flat line, so we know that we're measuring some sort of change in membrane potential. What we can say is that that change in membrane potential is, in ER potential is very fast. And so here's that voltage command step. I'm zooming in here. This is that change recorded by the electrode. You'll notice that the fluorescence change exactly tracks that of the membrane potential, or the electrode recorded change. And here's an image of the lunar ER staining. When we put burst on there as well, remember burst now goes to the plasma membrane. We can do a two-color experiment, and what I'm showing you here is that the plasma membrane potential becomes brighter, the ER membrane potential becomes more dim, less fluorescent. And so the direction of that ER membrane, that potential change, is the same as the plasma membrane potential. We have some control experiments showing that that Lunar Rover is flipped relative to our typical orientation, and that response is fast. The last thing I want to point out is that we were really surprised by this. It's not linear. At these potentials here, so change in fluorescence versus membrane potential, we have a steep slope here at hyperpolarized potentials and a shallow slope at potentials above 0 millivolts. So some sort of rectifying behavior. But we know that Lunar Rover is linear on the plasma membrane, and so this is something that we're really interested in exploring more. It seems to be there's some sort of mechanism for opposing changes to the ER membrane above 0 millivolts, and that's something we're looking forward to exploring. And with that, I'll just thank my group, and thank you for your attention.
Okay, thank you, Evan. The next speaker is Zev Gartner from UCSF.
All right, very excited to be here. Many thanks to the organizers for a chance to participate. And my lab's really interested in tissue structure. So we ask questions like, how does tissue structure form through self-organization? How does it break down in disease? And we hope that the answers to these questions will help us engineer tissue structure for a whole variety of different applications. All right, so what is tissue structure? We like to define tissue structure as the number of cells in a tissue, the cell type composition, as well as their shape and three-dimensional arrangement in space. And so a textbook example of the structure of one tissue, the mammary gland, is shown here on the bottom. So this is a branched ductal organ. And one type of cells, the luminal epithelial cells, shown here in green, I call them LEPs, line the lumen of the ducts. They're surrounded by a layer of myoepithelial cells, shown in magenta. And then finally, the entire thing is encased in a specialized form of ECM called the basement membrane. So this is the textbook description of what the mammary gland and other tissues in the body look like. But when you actually look at a tissue, what you see is this. So it's really quite heterogeneous. So you can see that each of these ducts have a slightly different size. They can have different shapes, different cell compositions, et cetera. And so if you really drill down and start to ask quantitatively how these different measures of tissue structure vary across different individuals and across different regions of a tissue, I think this really comes into focus. So this is a recent study from the lab where we looked at the tissues from 28 premenopausal women. And if we just quantify the composition of these tissues, what we found is that the composition of these three cell types is incredibly heterogeneous. That was surprising to us. We can similarly look at the arrangement of the cells in the tissue. Here we have to actually choose a quantitative structural metric. So in this case, we asked what fraction of those green luminal epithelial cells contact the basement membrane. And again, this is just looking at the tissue from a single donor. What we see is that at different regions of the tissue, that structural metric is once again highly heterogeneous. And if you look across time, we also know that tissue structure can change across developmental timescales. But if you look at very short timescales, what we can see is that different regions of a tissue can have very different dynamics. So in this case, out at the end of the ductal tree of a mouse mammary gland, cell shape as well as the topology or the connection between cells can be incredibly variable in time across tens of minutes to hour timescales. Whereas in the same tissue out along the duct, now we see cell shape is much more fixed and the topology is also fixed. Now, what's really exciting is that these properties of tissues seem to be intrinsic to the cells themselves. So we can take these cells or tissues out of the body, we can culture them in three-dimensional matrices, and they recapitulate many of these same properties that I just described to you. So this is an organoid from Andy Ewald's lab, and you can see at the invading tip of this organoid, the tumbling cells, the high cell dynamics, whereas at the edge, the cell's dynamics are considerably reduced. So how can we go about understanding what drives order or structure in tissues, and conversely, what can be driving some of this heterogeneity or disorder? Well, looking in vivo, we have a problem, which is that size, composition, cell arrangement, these parameters are all very closely coupled to one another, so they're going to be very difficult to study. And this is compounded by the fact that the microenvironment in tissues can be incredibly heterogeneous as well, and it's very difficult to visualize the dynamics. Luckily, we can take cells out of tissues, and we can use a variety of bioengineering tools to reconstitute organoids, and where we very carefully control for their size, for their composition, and for their microenvironment, and then we can use live cell microscopy to image their dynamics. And so this is a strategy that Vasudha Srivastava and Jennifer Hu decided to use to tackle this question of where does order and disorder in tissues come from? And I just found out that Vasudha has a poster, so if you want to find out more about this, please check it out. Okay, so Vasudha went about building many hundreds, thousands in fact, of these small tissues, put them on a microscope and imaged their self-organization. And so this is sort of a gallery, if you will, of some of these tissues after they achieve a steady state structure. So that you can see that the cells are moving around, they're dynamic. She put together a pipeline to segment these images and extracted out the same structural metric that I described to you a moment ago. So what fraction of these green cells contact the basement membrane? And when she quantified these data, again, across hundreds of tissues, this is the distribution that she saw. So what do we take away from this? Well, here's the most probable configuration of the tissue. So it has about 25% of these green LEP cells contacting the basement membrane. We essentially never see this structure with all the green cells on the outside. And we do see the textbook structure, but it's a minority of the structures that we see. So clearly there's order in this distribution, right? So this distribution is shifted to the left. It has 50% green and magenta cells. But there's also disorder, right? It's a very broad distribution. So where does the order come from? Well, there's about 70 plus years of research to really try to understand how cell-dense tissues like this one undergo self-organization. It's a process called cell sorting, and it's governed by cell-type specific differences in interfacial tensions between cells and between cells and their ECM. So you can think about an overall mechanical energy of the tissue, which is going to be the sum of the product of each type of interfacial tension times the total area that that interface has in the tissue. And the tissue tries to minimize this overall mechanical energy. We can use different biophysical techniques to actually quantify these interfacial tensions. These are the values that Vasudha measured. And it turns out that the principles that govern cell positioning in this tissue are actually really simple. And that's that the most energetically favorable interface is between those magenta myoepithelial cells and the surrounding ECM, explaining why they're on the outside. And the least favorable interface in the tissue is between the green luminal epithelial cells and the ECM, explaining why they're excluded from that interface. But this clearly isn't the full picture, because if this is everything that was going on, then we would actually predict there to be no green cells at the edge of the tissue in any of these structures. And that's because this mechanical energy gradient decreases monotonically as we remove luminal epithelial cells from the edge. And instead what we see is that the most probable tissue that we see is one with about 25% green cells at the edge. So what gives? Well, Vasudha had an insight, which is that if you perform a thought experiment where you take a tissue and you imagine having these dark and light gray sites, and you just start dropping your green and magenta cells into the tissue, the most probable configurations that you're going to see are the ones shown at the top, these sort of random looking configurations. Whereas the ones shown at the bottom are actually incredibly unlikely. There's exactly one way to put together two building blocks to give those two structures. We can actually show this experimentally by directing the self-organization of tissues, not with luminal and myoepithelial cells, which are mechanically different, but with two populations of, say, myoepithelial cells, one which we label green, the other which we label red. When we do so, the tissues still are very dynamic. The cells move around. They achieve a steady state structure. And when we measure that distribution, now what we see is a Gaussian distribution nicely centered at the composition of the tissue, which in this case is 50%. So from these experiments, we took a bit of a conceptual leap, and we said we have a mechanical energy gradient directing self-organization, and we also seem to have the impact of entropy on this distribution as well, right? Some configurations of this tissue are more probable than others. And so we asked, could we begin to model the ensemble structure of these tissues using Boltzmann statistics, using the tools of statistical mechanics? So we hypothesized that we could define this macrostate probability P sub I, which would be a function of the degeneracy of each macrostate, this gray distribution, which I showed we can measure, the mechanical potential, which I also showed that we can measure, this parameter beta, which you can think of as being the baseline mechanical fluctuations of the tissue, and it's sort of an inverse temperature. And finally, this entire thing is normalized by a partition function. And so using this concept, we can ask, can we begin to predict how changing these parameters, which you can manipulate by changing the microenvironment or cell biological properties, to predict how this ensemble structure would change. So for example, we can show that as we change the composition of the tissue, we can change the distribution. So nice match between theory and experiment. Similarly, if we change the size of the tissue, we can change the distribution. Again, nice match between theory and experiment. And finally, going in and manipulating the cells that are responsible for controlling this mechanical energy gradient also allows us to change the structure of this distribution. So just to wrap up, what we've done here is we've reconceptualized cells as living building blocks, similar to the way you conceptualize molecules and atoms as non-living building blocks. And by doing so, we can begin to predict and actually engineer the ensemble of structures that we see in vivo, beginning to explain what some of the fundamental drivers are for tissue structural heterogeneity. We now want to ask, what happens when we add cell-cell signaling into this mix? How can we think about how organisms control this distribution and narrow it? And finally, most interesting to us, is what can we learn about the loss of structure, the increase of disorder that occurs in diseases like cancer? So thank you very much for your attention, and thanks to all the people on the side here.
Okay, thank you, Zev. Continuing the contributions from UCSF, we have Bill Degrado.
Well, hello. Want to thank the organizers along with the others for this wonderful, wonderful meeting. We've been like drinking from the fire hose all this wonderful science and loving every minute of it, so thank you all. So can we go back one? Oh, I can do that. All right. Which way's which? Laser pointer, this one. How do I go backwards? Red? Okay. Well, the larger one. This one? Middle one. Okay. And this is backwards. Okay. There we go. All right, so I'm going to, I've lost a minute. I'm going to talk today about detection and recognition of conformational strains of tau and synuclein. And we're going to do this by spectroscopy as well as computational design. All the work I'm going to describe is in collaboration with the labs of Carlo Condello and Dan Southworth at the IND here at UCSF, directed by Stan Prusner. And following Jim's injunction, everything I'm going to tell you about is not published. He said it should also be interesting. You'll see whether it is or not. Now, I think we all know that basically a lot of organ diseases and neurodegenerative diseases are explained by the prion hypothesis, in which proteins aggregate in beta-rich arrangements, and then they recruit more protein and more protein, eventually forming amyloids, which are the end products. And the conformation we've learned of these final amyloids and presumably the intermediate prions depends on the disease. So if you look at tau that's come out of a CTE patient, it looks like this, as shown in Shear's lab. Here's what you would see for Alzheimer's disease, really quite different other than being beta sheet. And then here is Pick's disease, and on. So we'd really like to make genetically encodable tags and small molecules that really can tell us, oh, I've got this particular strain, and be able to do this in cells, in tissue, and in vitro. Before we do that, let's just think a little bit about how amyloid is formed. And you have a primary nucleation step, and then monomers are added and come on and off. And this is what we usually think about. But if you look at what contributes kinetically, fragmentation is huge. This is really a really important part of the overall process. Once you make fragments, they can now side on, they can nucleate more fibrils, and they can also nucleate a oligomeric species. And so somewhat counterintuitively, it might really make sense to stabilize amyloids. And certainly we want to block this secondary nucleation by binding to the sides. But how do you drug something that looks like that, huh? It's flat. It's not like anything you'd ever want to drug. Well, first we have to recognize it. And I'm going to tell you about spectroscopic probes to do that. And this will be done by Yang. Next, we're going to talk about the first structures at high resolution of a PET tracer, small molecule, first molecule we've seen bound to tau or any fibrils. And then finally, from Haley, we're going to put it together and design genetically encodable tags. Okay, so let's start with detecting amyloids. And many groups have screened through many dyes to see which stain amyloid. There's nothing new about that, but it's not very specific. On the other hand, they're all recognizing a different environment, and we thought that we could read that out in the spectrum. And so we just took a simple microscope, fluorescent microscope, that could do excitation and emission spectra. And we looked at 6 different amyloids. We played them all out, and we looked at hundreds of examples of each. And then we get spectra that look like this. Now, what you can do is to machine learn on all of the spectra to find out, is this a alpha-synuclein fibril of one type or of another type here? Is this A-beta of 2 different types? And we have different taus. And you can do this by just vectorizing each individual spectrum, excitation emission spectrum, and then you use supervised learning or unsupervised versions like PCA and UMAP, and that's what we're seeing here. So beautiful separation, great discrimination. We can also quantify the degree of separation that we see in this heat map. So these are for 4 different amyloids, and here these are the different dyes that we tested. And now we can select one for our project of interest. Here is like a brain slice. We're seeing tau tangles, beautifully separated and identifiable from A-beta plaques, shown here, simply using a single fluorophore. And most excitingly, though, we really have spatial resolution here, as opposed to what happens if you just grind up the tissue. And so we can see in Pick's disease that the tau inclusions in the neurons are conformationally quite distinct from those in the astrocytes. So that's just one example that we're developing and learning about. So now, what can we say, though, about how these dyes actually bind and other small molecules? And here, in Dan Southworth's lab, Greg Mertz solved the structure of a small molecule out of Genentech, a PET tracer, bound at about 2.6 angstrom resolution to disease brain amyloid tissue from Alzheimer's disease. And the first thing we see is that the site, somewhat surprisingly, is very polar. So it's lined by glutamine, lysine, and aspartic acid, but all of those side chains completely satisfy their hydrogen bond potential. And the apolar portion of the drug just matches perfectly with the methylene groups of those side chains. So it's perfect complementarity. Now, they also stack like this. And you see very effective pi-pi interactions. And by tilting like this, you can maximize the pi-pi interaction while matching the period of the amyloid. So pi-pi is 3.5, and then this is 4.7 angstroms. And so this is kind of a new principle, although it shouldn't be surprising if you know anything about Zev Gartner's work, that if you want to bind a surface, you have to not only think about binding, the free energy of binding of your molecule to the surface, but the monomer-monomer interaction. And this is really what gives you your positive cooperativity. And when you have a very, very polar surface, what can really provide the driving force for assembly. So now how can we use this? Well, let's now move to protein design. And let's look at synuclein. This is synuclein when bound to a membrane. It's in a helix, in solution. It's a random coil. And then it has many different conformational forms that have been studied by cryo-EM. And what we wanted to do was design a molecule that just binds to one of these. And we do this by looking at the most distinguishing feature here that isn't present in the others conformationally. So that's what we're going to do. We're going to pull that one out. And now we are going to design, in this case, a helical peptide with a fluorophore that's going to hopefully light it up. So how do we do that? Well, what we do is we excise this distinguishable region. This is the barcode we want to read. And so now this is it, now pulled out. And now we can take this barcode and look at its conformation. See, do I see that anywhere in the PDB? You do, and how? You can see here's one match. And in fact, we see about 3,000 matches, all with the same conformation, all giving us hints about how you might want to recognize this. Because we can say, hey, what binds against it? And to do this, we use Qubits, a program written by Nick Polizzi when he was in the lab. You see a huge mess, of course. But you can cluster them, and you can find which ones have the most favorable interaction with the amyloid, or I mean with our barcode there. And now, following Zev, we really have to think about not just how it interacts, but how each of these helices are going to interact with each other. That has to be highly designable and highly favorable. We do that by using flexible backbone design in Rosetta, using an algorithm that was sort of developed also by Nick Polizzi together. And we get nice hydrogen bonds and pi stacking to drive this as you would expect, and hydrophobic complementarity. So does it work? Haley made 4 different peptides. We're at reasonably early stage. What we've shown is that they, of those, only one lights up amyloid, and this is synuclein particles here. And we can also show that it's pretty specific for alpha-synuclein, the form that we wanted. And we can also see that we're still imaging down at about 1 nanomolar. Full biochemical experiments also showed that it's binding in the right stoichiometry. We don't have a structure yet. Okay, so, well, thank you very much. Let's see. And I'm over, so I better stop here. Thank you.
Okay, thank you, Bill. Our next speaker is Alice Ting.
Advancer, okay. All right. Well, thank you so much for inviting me to participate. My lab harnesses both natural evolution and directed evolution in the laboratory to develop molecular technologies that we hope are broadly useful for cell biology and neuroscience. So one of those areas is proximity labeling, which uses these promiscuous enzymes like Apex and TurboID to map spatial proteomes and transcriptomes and interactomes in living cells. Another area is the molecular editors that we're trying to develop to manipulate specific biomolecules and pathways in living cells. And then the third area are molecular recorders that convert analytes or transient cellular events into stable changes in the RNA of living cells. And so today I want to tell you about some unpublished work to record extracellular signals in RNA. Okay, so first of all, the motivation for developing molecular recorders in general, as we've seen in the earlier session, Evan Miller's talk and so on, real-time biosensors have been transformative for cell biology, but they do have limitations. They are limited to small fields of view and the study of just a handful of cells at a time. They're difficult to multiplex with other types of technologies like transcriptomics or proteomics. And it's also not possible to select out cells that have displayed interesting phenotypes using these real-time biosensors and then subsequently manipulate or reprogram them. And so we have over the years become interested in molecular recorders as an alternative to real-time biosensors that can enable that scalability and multiplexing. And so the general idea is to have some molecular tool that can convert a variety of different signals, whatever you might be interested in, into changes in the transcriptome of the cell or in a specific RNA reporter, however you wish. And then the RNA change can be read out either by RNA sequencing, or the RNA can encode a functional protein such as a fluorescent protein that enables microscopy, or channel rhodopsin that lets you go back and manipulate the membrane potential of a cell, or even a therapeutic antibody that's secreted at sites of disease. And so over the years, we've developed different types of molecular recorders. We started by building recorders for detecting elevations in cytosolic calcium. These recorders are also gated by light so that you have temporal specificity. And these calcium recorders, we've used them to stably tag transiently activated neural ensembles in the mouse brain and correlate them to specific behaviors. And then another type of molecular recorder is highlighter, which converts correct protein localization into a change in RNA. And we've used highlighter in combination with CRISPR screening to discover regulators of tail-anchored mitochondrial protein targeting. And so since the development of those types of tools, we've become interested in being able to sense also extracellular signals. And in a number of different ways. So either extracellular signal that's presented on the surface of a proximal cell, or a secreted soluble signal that can be recognized. And so, how do we think about developing recorders for this detection of extracellular signals? Well, first of all, there are already similar technologies in this space, most famously the chimeric antigen receptors that enable modular antigen detection with T-cell activation as the output. There's also the synthetic notch system developed by Wendell Lim's lab, also a very modular system that can detect various antigens and give transcription factor release and gene expression as the readout. One challenge with these two types of platforms is that they, by their mechanism of action, they can only detect cell surface antigens and don't work for the detection of soluble antigens. MESA is another platform that can detect soluble signals, but either the signal has to be homodimeric and induce this dimerization of this system, or you need two independent binders that recognize distinct epitopes on the same antigen in order to turn on the MESA platform. And so we've been interested in trying to think about a more general and versatile system that can detect all different kinds of antigens, including soluble or monomeric ones, using just a single binding module. And in addition, we wanted a system that has built-in drug control, so we can turn it on and off at will, that has a very versatile output, so driving transgene expression and not just T-cell activation, for example. And we would like the system to be very modular in its design, so that you can just replace the binding module with a different one, and it works without having to do like a whole engineering or evolution campaign. And so we selected for our platform a GPCR, so G protein-coupled receptors, which are experts at converting extracellular ligands into intracellular signaling. There are hundreds of them. They respond to a wide variety of signals. They undergo conformational change upon activation. However, they are completely not modular in the way that they recognize ligands. And in the past, to alter the ligand specificity of a GPCR typically requires extensive engineering or directed evolution, which is what we wanted to avoid. And so in order to get this to work, though, and meet the requirements that we had in mind, we had the key idea to start with a ligand-gated GPCR and to not actually try to alter its ligand specificity at all, but instead have a different strategy where we take a peptide antagonist that inhibits this GPCR and tether it to its extracellular end so that it's auto-inhibited. And then what we would do is introduce antigen gating of this peptide antagonist. So we're not using the antigen to gate the GPCR itself. Instead, we introduce this peptide antagonist and we gate that antagonist. And the way that we do that is by introducing a binding module adjacent to that peptide antagonist, such that the binding by an antigen sterically interferes with the function of that peptide antagonist, so that relieves autoinhibition. That's the idea. And then that means, if it's not autoinhibited, that the ligand can come in and activate the GPCR and turn it on. So in other words, this extracellular thing that we've built here is like a molecular switch. It can either bind to antigen or it can auto-inhibit, but it can't do both at the same time. And in addition, other features of PAGER, so the specific ligand or drug that we've selected is orthogonal, so it's not able to act on any of the endogenous GPCRs. And furthermore, to convert GPCR activity into gene expression, we tethered previous components that we've developed for this tool called Spark. Oh, wow, okay. Mistimed this. I haven't even shown you any data. In order to convert this into gene expression. Okay, so basically it works for detection of a model antigen, cell surface GFP, with really nice signal-to-noise ratio. It also works for the detection of soluble GFP, down to a KD of 10 nanomolar, which corresponds to the affinity of the nanobody that we used. And most excitingly, it's quite modular, because we could just swap out the GFP nanobody with binders against other soluble antigens, and PAGER worked on the first try without any optimization of geometry or linkers. And so we're pretty excited about this as a platform for very modular detection of antigens, both cell surface and soluble. And Matt Ravelin is a postdoc in the lab who is presenting a poster here. And one really cool thing that he's done is just by including a protease cleavage site here within this tether on the extracellular side of the GPCR, he's able to convert PAGER into a detector of protease activity. And so this is data showing that you get really nice signal to noise for detection of thrombin activity, And because that nanobody binder is still there, it's an OR gate, so it can also be turned on by antigen binding. And so we're pursuing some applications of pager. One is with Crystal Mackle at Stanford to use it in T cells to detect signals that are not accessible to traditional platforms like CARs and synthetic notch. For example, immunosuppressive signals like TGF-beta or IL-10, and then converting that into expression of CARs. And then we think there are cool applications in neuroscience as well. I'll end there and just thank Nick, who has been the amazing postdoc that has led the entire pager effort, and Matt, who joined more recently to help with pager and also to extend it to protease activity detection. These are the other members of my lab that I'm extremely grateful for, our sources of funding. Thank you for your attention.
Okay, thank you, Alice. The last speaker of this session is Peter Kim from Stanford.
Thank you. I also want to thank the organizers. I think this has just been a fantastic day, and really it's so much fun to be back together again. I'd like to give 3 lightning talks, if you will. The first is to talk about our second generation COVID vaccine. The overriding goal of this work was to solve for the problem highlighted here, and that is that low-income countries are simply not getting vaccinated. And even if one doesn't have a single humanitarian cell in one's body, Omicron demonstrates that it's in your selfish best interest to help solve this problem. Our vaccine is a protein nanoparticle created by fusing the SARS-CoV-2 spike protein to ferritin at the 3-fold axis of symmetry formed when ferritin forms a 24 subunit particle. The cryo-EM structure of this nanoparticle was determined by Kai-Ming Zhang in Hua Chu's group at Stanford. Importantly, we deleted the last 70 residues of the spike ectodomain because Colin Zamanek, Joe DeRissi, and Michael Wilson and their colleagues showed that these 70 residues, highlighted with the blue oval, are highly immunogenic in infected people, and that the antibodies that this region produces are directed against linear epitopes. And so therefore, they're not going to be neutralizing, and we wanted to remove this distraction from the immune system to the immune system in our vaccine candidate. And it turned out that this was important. In work that we are writing up now, we've improved the vaccine by introducing the so-called hexapro mutations. We've established a CHO cell line to produce the material in large amounts, and we've shown that we can elicit neutralizing antisera against all of the variants of concern in rhesus macaque monkeys, using only alum as the adjuvant, which opens up the door for making a pediatric-friendly vaccine, including immunizing infants. A manufacturing partner has been secured, and the prospects for making this vaccine at very low cost look very promising. And as you might imagine, a company has now taken over the development of this product. Next, evovelocity. This is the work spearheaded by Brian He, an extremely talented postdoc with machine learning expertise. He uses protein language models to predict the directionality of changes, of amino acid changes between 2 protein sequences within evolutionary trajectories. And then, based on the local predictions, he predicts the flow of evolution across the network. And that flow is what he calls evolutionary velocity, or evovelocity. And his first paper in Cell Systems showed that this works for many different protein families, including, as shown on the left, on the bottom, the evolution of influenza virus from 1968 to 2007, and, as shown on the right, for cytochrome C, where evolution is mapped over geologic eons. But while this work shows that protein language models can predict evolution retrospectively, he wanted to know whether or not they could also run evolution forward to prospectively design new proteins. And the answer is yes, as he's shown by doing this for 7 different monoclonal antibodies, 4 of which were approved by the FDA. In the example shown here, by testing only 21 variants of a monoclonal antibody called C143, he improves binding to the spike protein and improves viral neutralization up to 30-fold. I want to emphasize that these models are completely unsupervised. They're trained on all types of protein sequences, not just antibodies. They have no information about the target antigen, binding specificity, or protein structure. The algorithm only requires a single wild-type protein sequence and computes the amino acid changes in less than a second. Moreover, using previously published fitness measurements from high-throughput scanning mutagenesis experiments, Brian has shown that the same language models can guide efficient evolution for proteins where fitness is determined or is defined as antibiotic resistance, cancer drug resistance, enzyme activity, or viral replication. The big question, of course, is why does this method work? Fundamentally, we think that these results actually challenge the prevailing notion that evolution is random. Instead, we hypothesize that evolution somehow remains on a manifold in which a substantial portion of mutations that are predisposed to have high fitness. If that's true, this raises the very interesting question of how natural evolution actually occurs on a manifold. Finally, recon abs. Starting with the flu. Upon influenza infection or vaccination, the antibody response is overwhelmingly against the head region, as shown here, of the hemagglutinin protein, or HA, which is therefore considered immunodominant as compared to the stem region. Amino acid residues on the head change continuously, which necessitates the production of new vaccines against influenza every season. But rarely, antibodies against conserved epitopes on the HA stem have been isolated. And some of these monoclonal antibodies have very broad and potent neutralizing activity against a wide range of influenza viruses. The spike protein of SARS-CoV-2 also has a highly variable head region and a stem region with highly conserved epitopes. Shown in red are mutations that occurred in Omicron. Shown in blue are some highly conserved regions on the stem. And the green oval down at the bottom highlights that many residues are 100% identical across approximately 40 different coronavirus spike proteins depicted here. Early in the pandemic, we, and many, many other people, sought out to identify monoclonal antibodies that would bind to the blue, highly conserved epitopes. And the hope was that these antibodies, or at least some of these antibodies, like in the case of influenza, would be broadly and potently neutralizing and would therefore serve as useful therapeutics as the virus evolved to these different variants. Eric Waltery at the BioHub used bioinformatics to identify candidate human monoclonal antibodies that were predicted not to bind the RBD, or receptor binding domain, because we weren't interested in that, on the spike head. Peyton Weidenbacher and others in the lab were able to isolate and epitope map non-RBD binding antibodies that bound to both SARS-CoV-2 and SARS-CoV-1, indicating that they bound to highly conserved coronavirus epitopes, presumably on the spike stem. But none of these antibodies were neutralizing. Others were also not able to isolate stem-binding antibodies with potent neutralizing activity. So their antibodies, like ours, went into the garbage can. But then Peyton came up with the idea of receptor-blocking, non-neutralizing antibodies, or recon-abs. And the idea was to use the non-neutralizing antibodies that bound to highly conserved stem regions and tether them to the ectodomain of ACE2, the receptor for SARS-CoV-2. The non-neutralizing antibody would ensure binding to any variant of CoV-2, and the tether would hold the ACE2 domain at a high local effective concentration where it could act as an inhibitor. So he designed those constructs with a TEV protease site so that the control experiment could be easily done. And sure enough, the recon-abs showed broad-spectrum neutralizing activity against all variants, but when the TEV protease was added, activity was lost. The most advanced recon-ab to date is shown here, which contains two non-neutralizing antibody combining sites tethered to an ACE2 domain. As shown in the figure, it shows sub-nanomolar inhibition against all viral variants, as shown in the yellow symbols, but the activity goes away upon TEV treatment, as shown in the green symbols. In future work, we're interested in using this strategy to more broadly prepare a parts box, if you will, that could be used to assemble an appropriate recon-ab when a new pandemic strikes. Thank you for your attention. This slide gratefully acknowledges the funding agencies and my terrific laboratory.
Okay, awesome. So I think we'll take questions for the speakers in turn. So the question for Paul is, do you think your approach to reducing viscosity for bispecific antibodies could be translatable to other biologics?
So I described a couple of approaches. One was using excipients, and certainly that's very widely and commonly used for sort of biologics. Mutational approaches, I think the answer is yes. Exactly what mutations? I think it's more than just aromatics. Things like removing charge patches and hydrophobic patches can be important too. The tricky part is doing that and also maintaining function and also not just inadvertently making your molecule immunogenic. So I think in principle the answer is yes.
Do you want to comment on how you would do those two things at the same time?
I think medium to long term, I think it's kind of potentially a great application of machine learning because it's kind of a multi-parameter optimization. I think at the moment our kind of understanding of factors impacting immunogenicity are fairly rudimentary, and I think we'd be kidding ourselves to do that at the moment.
Okay, thank you. Okay, for Evan, what properties make for good voltage-sensitive fluorophores? In other words, why FITC variants as opposed to coumarin, cyanine, or Bodipi?
I think it depends on the biological question, what observations you want to make. We start with the xanthines because we've got good synthetic methods to make them. You can do it with, in principle, those other fluorophores as well. Coumarins maybe are a little bit blue, perhaps, and so you want longer wavelength things. A lot of the really great optical indicators, like GFP is widely used, and that's going to use up your blue and green wavelengths. So those are some of the considerations we think about.
Okay, thank you. Okay, for Zev, what are the factors that contribute to cell type-dependent variations in shear stress, and how do these factors differ between cell types?
So in thinking about the interfacial tensions between cells, there's a number of things that contribute to those. The one that we classically think about is the expression of adhesion molecules. But sort of the modern view of how self-organization works is that those adhesion molecules function by coupling to the actomycin cell cortex, where the sort of active forces are generated by cells. And so that provides at least two strategies that cells can use to modulate their sort of biophysical properties. One being changing the spectrum of adhesion molecules that they express, and the second being regulating the contractility of the actomycin cell cortex.
Okay, awesome. Okay, for Bill, do any of your dyes form capping motifs to prevent extension? And can you engineer capping proteins to work synergistically with your fibril stabilizing motifs? Yeah, sure. Do any of your dyes form capping motifs to prevent extension? Can you engineer capping proteins to work synergistically with your fibril stabilizing motifs?
Yeah, we haven't played with extension. Others certainly have. And let's see, there's a lot of natural proteins that bind side-on. TREM2 is one of the famous examples. So they certainly could be designed as well.
Okay, thank you. Okay, for Alice, how challenging is it to find autoinhibitory peptides to tether to your GPCR? Are such peptide antagonists known or rationally designable for orphan GPCRs?
That is a big challenge. All the data that I showed was based on one GPCR scaffold, for which we do have an antagonist peptide. There are natural peptide-gated GPCRs, and it would be easier to either find or design peptide antagonists for those. For other types of GPCRs that only have small molecule ligands, it's more challenging. Some of them do have toxins, natural toxins that bind to them and antagonize them, and so one might be able to envision creating a peptide antagonist with some portion of that toxin, and then utilizing computational design strategies, directed evolution approaches to get a good system.
Okay, and finally for Peter, how did your general purpose language model compare to supervised machine learning models that were trained for specific fitness prediction tasks?
Thank you. The referees have asked us that question as well. So if you're the referee, we've answered the question in the revision. And the answer is actually, well, to me it was surprising. The supervised models that are trained on antibodies actually do worse. And there are different interpretations or speculations as to why that may be. But the result is that they do worse. And of course, they also would not be expected to be able to apply to these other protein families either.
Okay, well, thank you. So we're actually not done yet. We have a fantastic panel coming up, so I think we should all thank all speakers in this session. And I'm going to... And then I'm going to turn this back over to Jim.
Okay, I'd like to, you've been hearing mostly just straight up science talks today, but there's a lot more to what we do than just the science itself. And we also have a lot of things going on in the industrial sector, for which we're really grateful to have three panelists to join us today. So I'd like to invite to the stage Ray Deshays and Shiva Malik and Andy Chan. Bill. Great, so I've crafted some questions myself to get things going, but if you have ones from the audience, if we have time, we'd love to have you put them in the chat and we can try to get to those. So let's see, Andy, there you are. Okay, great. All right, let me first, just a brief introductory comments. Ray Deshays is Senior VP of Global Research at Amgen, where he leads the team responsible for identifying the most innovative technologies and targets in drug discovery. Prior to joining Amgen, Ray was a professor at Caltech, where he pioneered a number of really important discoveries, most notably in the ubiquitin proteasome system. And one of the practical aspects of that was his development with Craig Cruz of Protax. He's a member of the National Academy, the AAAS. He received his PhD from UC Berkeley, so he's a Cal Bear. Woohoo! So Ray, I'd like to ask you, maybe you could just spend a couple minutes telling us about what excites you most about your position at Amgen and what concerns you the most there.
So I would say what excites me the most is the potential to influence the development of the next generation of medicines that can, be used to deliver enhanced value to patients. We have so many diseases where, there really are very few, if any, therapies, or the therapies that exist have all sorts of problems associated with them. And so the potential to really develop medicines to improve patient lives is enormous. And all of us have relatives, sisters, brothers, mothers, fathers, grandparents, children who've suffered from disease and have not had access to medicines that would help cure them or modify their disease. So that's the exciting piece. I think there's a lot of potential there. I'm particularly excited about the potential for making multi-specific medicines of various types, including but not limited to targeted protein degraders. I think we heard today there's all sorts of classes of multi-specific medicines that one could conceive of, and I think those really have the potential to bring us to unprecedented levels of efficacy while being very well tolerated by patients. What am I worried about? I would say that probably falls in more the general area of policy, especially related to some of you may be familiar with the Inflation Reduction Act. I think there's a lot of concern right now in the industry about what impact is that going to have on incentives for developing different types of medicines. And the thing that worries me in particular is it going to drive behaviors in a way that may optimize for industry but not optimize for society? So, for example, one of the biggest impacts on society remains cardiovascular disease. It's a very difficult disease area, typically small molecule therapeutics, and it's an area that arguably is underinvested in by industry given the societal impact, just because it's perceived that it's more difficult to be profitable in that area. And some of the changes that are afoot now may only serve to exacerbate that. So that's something that I'm concerned about.
Thank you very much for that, Ray. And next, Shiva. Shiva is currently Global Disease Area Head of Oncology at the Novartis Institutes of Biomedical Research, otherwise known as NIBR. Before that, she was 15 years at Genentech, where she was Vice President of Discovery Oncology. She's made some very important contributions to, in her scientific side of things, to the paradoxical aspect of RAF and RAF signaling, that sometimes you can bind with drugs and actually activate the pathway. She figured that out and then developed ways around it. She also is a product of the UC system, although as an undergraduate at UCLA, and then a master's degree from UC Irvine and a PhD from UCSD. Shiva, so same question to you. What excites you most about your new position at NIBR?
Thanks, Jim. So really happy to be here. And just like Ray, really doing something meaningful for patients is at fundamental level what I'm most excited about and why I'm there, why I was at Genentech for 15 years. And I think every single person at that organization has that same mission, and it's very personal. It's not just a job. I think just like probably all of you in the room here. But I think from a day-to-day feeling, what I find really exciting is the opportunity to stitch together fundamental biology, cancer biology in our case here, with really exciting breakthrough technologies, with data, and use that information to advance therapies to patients. And then what's super, I think, exciting and fulfilling is to actually see that science play out and understand it in the context of human disease and learn from patients and actually iterate on that and bring that back to the lab and figure out where we go next. I mean, I think the story of paradoxical activation is a terrific example of that. And I think drug development is really about playing that long game. It's taking these fundamental discoveries, turning those into medicines ideally, understanding what happens in patients and learning from that and coming back, because ultimately we're not just trying to delay progression of disease, right? We're trying to cure disease. And so all those elements are really required for us to reach that goal.
And what concerns you the most about it?
So exactly that the challenge, I guess, two main things. One is we're still not curing disease. We have made some significant advances, but really how do we get to cures, right? That's the kind of stuff that keeps you up at night. And then the second is, despite all the breakthroughs that we've had, scientific discoveries, drugs being approved, right? We're at a rapid pace of drug approvals. If you look at our success rates, that has not improved. In fact, it's gotten even harder. So I think the number everybody knows is we have about a 10% success rate, right? So meaning we have to give many, many attempts of taking molecules into the clinic before we actually get an approved drug. And some of those drugs just give you incremental improvements in disease. And so how do we improve that process, both improve our success rate as well as deliver better medicines for patients that are better tolerated, that really have more profound efficacy? And that's going to require actually understanding disease in patients, I think, right? And more data actually from the clinical side, translational science, in addition to the fundamental work that I think everybody here in this room is doing, as well as industry. Yeah. So.
Thank you very much, Shiva. So next we have Andy Chan. Andy is currently the Senior Vice President of Biology, Research Biology at Genentech, where he's been for 21 years. He's been part of the heyday of when Genentech developed all of those amazing therapeutic antibodies. One of his major contributions is understanding protein tyrosine phosphatases and how they impact T cell function recently, and in the past has found some interesting phosphatases that inhibit T cell function, and if they were targeted could probably unleash a horde of T cell activity that would be useful for cancer. He was previously an associate professor of medicine at WashU prior to joining Genentech, an investigator of the Howard Hughes Medical Institute. And then he moved to UCSF. So he is a product of the UC system too, as a associate clinical professor. He was an undergraduate at Northwestern, got his MD, PhD from Washington University in St. Louis. He received a number of numerous awards, including distinguished alumni from both of his schools, which is really saying a lot. Usually when people go, they sometimes just forget about you, but not Andy. So that's awesome. So Andy, what excites you most about the work that you're doing at Genentech?
I think this is always an interesting time in the intersection of science and medicine. What we've even just this afternoon, I had the pleasure to be able to hear the talks. The pace by which different aspects of science and technologies are advancing are actually phenomenally tremendous. This provides us new tools by which we can interrogate new biologies to screen for new compounds. as well as to be able to understand and interrogate human biology to a far, far more precise aspect. And all of these particular technologies have a unique potential to be able to either help us diagnose, treat, or as well as prognostics in human diseases. And now, I think further, it's the intersection and interplay between the different disciplines that likewise is also accelerating the potentials of each one of the individual discoveries at an alarming pace. So I think the fascinating thing in drug discovery and drug development right now is that the science is absolutely impressive. And the question is, how do we best apply the multitudes of advances that are ongoing into our drug discovery, target discovery, and drug development aspects? It's a very exciting time.
So, with all of that, what concerns you the most?
Yeah, in addition to the things that Shiva and Ray have already alluded to, I think one of the major societal issues that we face is how do we afford it all? Peter talked about the low-income countries that are not able to even get very cutting-edge technologies that have significant scientific clinical impact in the first world. How do we ensure that we maintain the incentives to allow innovation to occur, and yet allow these phenomenal discoveries to be able to help everyone, not just the first world?
Yeah. Thank you so much for that. I'd like to now sort of drill in a little bit deeper, asking the question. We'll start with Ray. Can you describe the 3 areas, disease and/or technology, that you think will be the most important for your company in the next 10 years? Now, maybe that 10 years is a little bit daunting, a horizon, given how rapidly things change. So if you want to back it off to 5 years, that's fine, given the rapidity of discoveries and how they're being advanced. So what 3 major areas do you think are going to be critical for Amgen in the next 5 years, let's say?
5 years is way too short. I think you have to talk about more than 10 years.
All right, go ahead.
We just had this thing where we met with Wall Street earlier in the year, and we can project out, roughly to 2030 based on the existing portfolio. But, my organization is all about what happens after 2030. So I would say, I'm not sure I'm going to give you 3. I would probably put near the top or at the top of the list the ability to target medicines to specific tissues as being of extremely high importance. And so, for example, just one example. It's now known how to get an siRNA to the liver by attaching GalNAc to it, right? If you could do that with every different organ system and tissue in the body, you could then use siRNA to treat a whole range of diseases that you can't go after right now. And so I would say addressing that problem and solving that problem, I think would be a huge advance. I'll say a second advance, we're not in the area of neuroscience, so this is not an Amgen specific thing per se, but we may get back there in the future, would be solving the problem of the blood-brain barrier. It's one thing I noticed when I first got to Amgen. If you just looked at the industry over the past 30 years preceding, neuroscience drugs used to be among the best-selling drugs in the world. Then they went generic, and they were, all the top-selling drugs in the world were not in neuroscience any longer. Many of them were antibodies. We have all these neurological diseases that exert an enormous toll on society. But many of the receptor systems in the brain, you have multiple very closely related family members that are very difficult to distinguish from each other with small molecules. if you could bring the specificity of biologic therapies to bear, the potential to deal with some of these diseases would be tremendously advanced. Yet, you know, with the existing technology, you simply can't get enough antibody in the brain to modulate targets appropriately in most cases. So if that delivery problem could be solved, I think that would have enormous potential. And I would say the third would be something I alluded to earlier, which is perfecting the science of multi-specific molecular medicines. You know, so if you look at our pipeline, you look at other companies' pipelines, there's a broad emergence of more complex modalities, which would include, you know, bispecific antibodies, trispecific antibodies. We heard about that in the last session. Mixed modalities like antibody-drug conjugates, targeted protein degradation, you know, small molecule chimeras and so forth. These have a lot of potential, but I can tell you from experience, there's a lot of difficulties with developing these types of medicines. And so I think a lot of new science is going to have to be invented to overcome the challenges posed by these and to realize their potential benefits for patients.
Ray, thank you very much. Shiva, same question to you. What are the sort of 3 areas, or however many you feel are really up there in the next 10 years, let's say, for Novartis and NIBR, to make the company really, you know, succeed?
Yeah, so I would definitely have to agree with what Ray said, which is tissue-specific delivery. And one of the technology platforms that I think is pretty exciting at NIBR is the radioligand therapy platform. And again, this is all about delivering basically radiation to every single tumor cell in your body. So it has pretty profound efficacy when it works. But the other aspect of it that I think is fascinating is that you can image patients first. So there's a theranostics piece, and we heard a bit about imaging here and how important that is. And I think that's going to continue to be this diagnostics piece, imaging piece, and basically this idea of you treat what you see, right? And so I think that's definitely an interesting and exciting new area, and it opens the door to different ways to probe tissue-specific delivery beyond antibodies and VHHs and other biologics. So using peptides and other modalities to get at tissue-specific delivery, and opens the door to completely, vastly different payloads beyond what we've seen in the past, like siRNA or cytotoxics, but now, you know, radiation, perhaps there's other payloads that you could envision delivering in a tumor or a tissue-specific way. The other technology that I think is really exciting is, of course, cellular therapies, both for cancer and outside of cancer indications. I think there's a lot of potential there. This whole notion of living drugs, I think, is really interesting, and combined with some of the novel engineering technologies, barcoding approaches, you could envision there's a lot you could do to actually develop living drugs, per se, that evolve and adapt to an evolving disease. So I think there's a lot of potential there. Now, the challenge with cellular therapies, of course, is the delivery, right? There's significant challenges. It's difficult to make it broadly accessible, obviously. And again, I think there's multiple different approaches that are being taken right now to break open that aspect of it. It's a technology hurdle. But from a value proposition for patients, right, that's where we're talking about long-term responses, potential cures, that I think is where we would like to be. And then the third thing is, you know, while NIBR isn't necessarily a diagnostic company, but I think early detection diagnostics is going to be really critical for us in, again, making significant improvements on disease. I think we need to have a really good understanding of early detection and then monitoring disease as it progresses, and doing that in as little as interventional type of approach, right? So we're able to do that, and having as much human data, clinical data as possible to understand both disease initiation and progression. I think that's going to be really a critical technology to develop.
Great. Okay, well, Andy, what about you? What do you think at Genentech and in your area are going to be really exciting, critical things to develop?
I thought Shiva was not going to talk about oncology. No, I'm kidding. Well, you know, Genentech presently is very, very invested in neuroscience. You know, neurodegeneration is a major cause of morbidity as well as mortality. And as Ray noted, you know, being able to deliver drugs very efficiently across the blood-brain barrier, as well as cell-specific targeting in the CNS are both very, very critical. The second area that I'm actually quite enthused about is that, and we haven't discussed this, is the area of regenerative medicine. I think there are a multitude of applications. This is still very, very early, and likely will also require things that Shiva brought up, which is our ability to be able to manipulate cell systems, living organisms, to be able to perform and sense the things that we want to do in the appropriate microenvironment. And the third area is an area that is very broad, but that's actually of preventive medicine. I think preventive medicine has probably had the most impact of early diagnosis, whether we can find infectious causes of subsequent malignancies or other more severe diseases. And this is a underappreciated and underinvested area.
Yeah. I was going to actually ask you about that because, you know, the focus of most pharmaceutical companies is to cure a disease that's already progressed. You know, you're already in the middle of it. Often you're too late. What do you think about that business of preventative medicines? I realize the clinical trial for something like that would be incredibly delayed, but what is the horizon? Do you think there's ways to predict who's going to get disease in very effective ways? And what are your I'm interested in all of your guys' opinion of that.
Well, that is my hope. Okay, I think there are clearly diseases where that have been well studied, where good and reasonably timed clinical studies can be performed, right? HSV vaccine, and COVID vaccine, infectious disease vaccines in well-studied areas have clearly been successful. I think in broader areas, it does require we in the industry, to be able to have long-term commitments with academic institutions to be able to understand what the natural progression is of diseases, and universities and academic centers have the patient populations where if there's a commitment in both parties, or even of NIH-sponsored studies, that we have to make headway to be able to understand what are the early biomarkers, whether it be Alzheimer's, whether it be cancers and the like, to be able to identify those biomarkers or imaging modalities, so we can do some of those earlier studies. There are many situations where that has happened. We just have to be able to make further commitments in this regard.
Shiva, so just following up on the preventive aspects, and as Andy mentioned, vaccines is the classic area of prevention. What do you think the horizon looks like for cancer vaccines? It's a very old field, I realize that. Do you think that's progressing at a rate that in 10 years that we might be able to take vaccines to prevent cancers?
Yeah, I mean, I think there's been significant improvements in the area of cancer vaccines, very early vaccines available for children in preventing cervical cancer. So there's been quite a bit, and there's a lot right now in clinical studies. But I think the challenge, again, remains the diagnostic piece. Is running those clinical studies are challenging when it comes in a prevention setting. And so I think not only do we have to advance cancer vaccines, right, there's multiple approaches right now being taken for developing cancer vaccines, but also in parallel building diagnostics to understand which individuals are predisposed to cancer or will develop cancers, and running clinical studies like that, right, interventional or prevention studies rather than intervention studies, which is largely what the cancer vaccine field is doing, where it's all, you have cancer and then you're given a clinical cancer vaccine. So yeah, I think the diagnostic piece is going to be really important, and then partnering with the FDA, because running these clinical studies that we're talking about, these interventional studies, are far different than, the endpoints are different, how you might execute them are different, so that will also be a requirement, I think.
Thank you. Just to change gears a little bit and sort of throw it out here to the audience, what do you think academics can do? Most of the talks you heard today were from academia. What do you think academics can focus on that will really accelerate those visions that you guys have? Ray, maybe you want to take the first part of that? How can academics participate?
You know, I think what industry is very good at is, you know, once the contours of the problem are understood, to then engineer a solution to the problem, right? That's, I think, where the real strong suite of industry is. Where industry is not as good, this is purely my opinion, is in defining the contours of the problem. And that's where I think academics can really excel, right, in sort of the open field, open space biology of identifying new problems and defining the contours of the problem, so that one could begin to then ideate about what the solution is and then engineer toward a solution. And of course, there's a gray zone in between those two, you know, and that gray zone is constantly shifting. But, you know, I think defining the biology and doing, you know, or in chemistry, defining the novel reaction mechanisms, you know, that'll allow you to build new types of bonds. Those are the things I think that where academics can really add a lot of value to what we do.
Are there some academic collaborations that you could mention that you have that are really about doing that?
Well, one example off the top of my head is we have a collaboration with the Institute for Protein Design, run by David Baker. And you know, the thing we're doing there is we're asking questions to see if we can come up with a proof of concept, okay? So just to give you one example, imagine, you know, we heard today about bispecific T-cell engager molecules in at least one of the talks that bring a T-cell to a cancer cell to kill the cancer cell. And a real limitation there is that there are not that many solid tumor antigens that are truly tumor specific. Most of them have some low-level expression on normal cells. And with that mechanism, if you have low-level expression on normal tissues, you've got a problem because you'll have on-target toxicity. So, you know, IPD has come up with a number of approaches, one being their so-called locker technology, for example, where you could think of ways of building various types of AND gates, you know, which would impose two criteria, not just one criterion on the cancer cell that it has to fulfill in order for the attack to happen. And, you know, so coming up with new ideas, new approaches for doing that, you know, I think that's a great example where they can go and do the blue sky work and say, oh, you know, we have 5 ideas, we're gonna whiteboard these 5 ideas. And then if one of them looks promising, you know, then that's something that we at Amgen could jump on and you know, engineer every possible conceivable permutation to come up with the one that is the perfect medicine.
Shiva, what about you at Novartis, NIBR? How do you think academics can better interface with the group there to accelerate your research and development?
Yeah, so we have a number of very large academic collaborations, including actually one with UC Berkeley and Dan Nomura. I think Dan was here, I saw him. Yeah, so that's a very large collaboration, and the second one being one with the Farber that's been ongoing for many, many years at Novartis. I think both those, I go back to what Andy said, it's the interface of disciplines, I think, that opens the door to a lot of innovation when we have these collaborations. So oftentimes, the academic labs that we're working with are bringing a skill or a know-how insight that we don't have or come from a completely different discipline. And it's complementing that maybe with what we may have internally, be it applied drug discovery or other aspects of the drug discovery process that may really complement what's going on in the academic lab. So it's really that interface that I think is powerful. The second aspect of academic collaborations that's been really impactful has been the opportunity to do more translational work. So we have some really rich academic collaborations where we are doing things like trying to understand disease in clinical studies in patients, getting access to patient data, working with our collaborator, where we may provide the molecules, but they're actually studying the molecules in patients, and having a very open, collaborative environment where we're both taking a look at the data, they're bringing insights, posing questions, and we're doing the same. So I think those have been really, the translational collaborations have also been really rich and fulfilling.
Andy, what about you at Genentech? What are you guys doing in terms of academic collaborations, and where do you think there's room to grow on the academic side?
Yeah, I think there are always wonderful opportunities for Genentech to collaborate with the academic colleagues on specific projects. You know, I think usually in industry we have phenomenal depth in the few areas of focus that we have. But in the end, it's always the innovative ideas that are more broad that we don't necessarily have the efforts at that particular time. So new ideas reach out to the industry colleagues and thereby initiate a actual project that we may well start small, medium, and then may well be able to grow larger. I mean, I think we've benefited, having been in the South San Francisco area, to have, you know, we have a joint postdoc program with Stanford. We have a large initiative with the Weill Neuro Hub that spans UC Berkeley, UCSF, and now the University of Washington. There are multiple different opportunities.
Thank you. Now, for the graduate students out there thinking about, you know, their next step after they graduate, many will be looking at potential postdocs. And each of your organizations have postdoc programs. I'm most familiar with the one at Genentech, which is one I'm closest to. But Andy, why don't we start with you? Why don't you tell us how the Genentech postdoc program works, and what is kind of the, you know, how many do you take in, and what is that like, and why should they think about that versus a postdoc in an academic lab?
Yeah, so we're very fortunate, you know, to have a long-standing postdoc program that started with you.
It's grown a lot.
We have about 140, 150 postdocs presently. Our postdocs are not meant to be entry positions into Genentech. They're meant to be independent positions that are not overseen by the company. They're actually asked not to work on company projects. They're supposed to do basic science discovery. And the goal of our postdoc program is to get our postdoctoral fellows to be in as competitive a position as possible so they can go back out and get an independent position, whether it be in academia or industry. The one thing I would tell, when folks apply to the Genentech postdoc program is that you should only honestly come to Genentech postdoc program if you have an interest in industry and/or if there is a specific technology that we have in-house that you believe you really want to take advantage of for that specific project. Ultimately, if you want to be an assistant associate or full professor and have little to no interest in industry, I think you should seriously consider a postdoc in the academic area.
Okay. Shiva, do you want to talk about the program at NIBR?
Yeah, I'm happy to talk about that. So there's actually 2 tracks for postdoc programs at Novartis or at NIBR. There's the innovation and discovery track, and they differ actually pretty significantly. So one of the tracks is really about learning drug discovery, and so postdocs in that track are actually involved in project teams, and your record of accomplishment during that period may be more about patents rather than publications. The second track, which is more discovery, is actually more of a traditional postdoc where your record of accomplishment is publications, usually around areas of science that various lab heads at Novartis are interested in. I don't think it's as big as the Genentech postdoc program. I don't believe there's over 100 postdocs there. I think it's a little bit smaller. But it's a really active program. There's a great postdoc community. We have many postdocs in oncology. Actually, they attend all our town halls, our meetings, so they, even regardless of what track you're on, you would get a very good sense of how industry works, how project teams work, how the science and drug discovery works at a company. So I do think if you're, like Andy said, if you're interested in a career in industry, it is a great way to learn about how drug development works in the industry setting.
Okay. Ray, do you want to comment on the Amgen?
Yeah, I'll just echo some of the things that have already been said. We have a much, much smaller program than Genentech has. I think we have 35 postdoctoral fellows. They are mainly based either in Thousand Oaks or up here in South San Francisco. The program was just taken over by Ryan Potts, who I had recruited from St. Jude maybe 3 years ago now, and who's trained a number of postdoctoral fellows when he was in academia. And he's creating a series of I think what he's calling centers of excellence to try to have the postdocs working on, a particular range of topics to try to build up internal communities within that group of 35 people where, so there's an intellectual environment for the postdocs to engage with each other in their research. And, I would say when postdocs come in, we appoint them for 3 years with the idea that, it would be a short postdoc, but then there's an opportunity for them to get an extension beyond that 3 years. I would say it's a great, like Shiva mentioned, if you want to go into industry, it's a great way to come in and learn about how industry works. And, what we also try to keep, I would say, our postdocs, at least from my point of view, I think it's really important to keep them insulated from the pipeline, right? So that they're working on projects that are not pipeline oriented, so that they're free to be able to go out and talk about their research and publish their research, without concerns related to IP and so forth.
And then for the postdocs and graduate students, because many of them are taking positions directly, graduate students are taking positions directly in industry these days, and postdocs too, especially from the Bay Area universities. What are the things that if you had to, sometimes I will get asked, because I was in industry for a long time, you know, what are the skill bases that I need to have in order to be sort of appropriate? And so there's kind of this skill issue. There's also, though, the teamwork issue. There's also the invention issue. There's also the, you know, all of those other things. When you're interviewing what advice would you give folks about interviewing at your respective companies, and which factors do you consider to be the most important general ones? Do you want to start, Ray?
I'm trying to formulate my answer here. Maybe I'll answer the question I want to answer instead of the question that was asked.
That's fine.
So I get asked this, you get asked all the time about, you know, students or postdocs are thinking about making that decision about whether to go into industry or not go into industry. And the question often boils down to, you know, what's sort of the fundamental difference between those two? You know, why would somebody decide to go in one direction or the other? And this certainly comes up to the part of the question you asked about, like, you know, how does that influence how you interview for these positions? I would say there's two key factors that would define, you know, people who are going to come into industry as opposed to doing any of the number of other things you could do with a PhD in this field. And the first is a desire to make an impact on patients. You know, you see a lot of people in research. I loved doing basic research, but there are a lot of people who feel like they want to have more of a direct impact. And, you know, if you go into industry, you can participate in developing a drug that directly impacts on patients' lives. And I think, you know, some people, that's a very important driver for them. And, you know, that's a great reason to go to industry, because it's just your potential to have that impact, I think, is just higher. The other is a strong, and I think these two go together, is a strong desire to work as part of a bigger team. You know, as an academic and running an academic lab for a long time, although, you know, half of our papers we collaborated with other labs on, you know, you're charting your own course, you're doing your own thing, and, you know, you have collaborations, but it's not quite the same, right? You're really blazing your own path, so to speak. And if you want to accomplish something like developing a drug, I mean, I think developing a drug is the hardest thing human beings do. You know, I always tell my people at Amgen, this is not rocket science. You know, rocket science is easy. You can calculate to a high degree of precision where a probe is going to land on Mars, like, you know, 18 months from now, or however long it takes to get to Mars. But you can't do that with drug development. And so you need very large groups of people working together cooperatively in teams. And, you know, there's a real sense of satisfaction of working with peers to achieve a common objective. And I think that's another thing that really characterizes people, in my mind, who want to go and are going to be successful in industry.
Shiva, would you like to answer that question?
Yeah, so I thought that was really well put on what we're looking for, but maybe I can give some practical advice in terms of, I review lots and lots of CVs, and I can tell you, coming from an institution like UC Berkeley or UCSF will definitely merit a very close look at your CV. These are
Stanford too?
And Stanford, sorry, any of these Bay Area institutes for sure. No, that's, of course.
And San Francisco State too.
And San Francisco State. So definitely your record of accomplishments, work on your CV, make it crisp and clear so that it's obvious not just what your papers are, publications are, but what your research findings were. But really the most critical thing is being able to articulate that, communicate that really well. And it shouldn't just be telling a narrative of, well, I did this and I started that and going on, telling me a narrative. But first of all, you should be able to articulate why what you did is important. Why should I care? Why does anybody care? Why do you care about the project that you did? You should really be able to articulate that. What the impact of that project was and how it shaped the field. So how did it leave things? What direction? What new insights are left now? You'd be surprised. It's very difficult to do. Even if you have Cell Nature Science papers on your CV, so clearly high impact work, it's very difficult to articulate those types of things of why the work you do matters, why it's important. And I think candidates that I see that do that really effectively just stand out. And it requires you to think really, really hard and deep, right? Stepping back from the details of the experiments that you did and asking broadly, what did what I do, like how is it meaningful? So really think about that, and actually taking the time to talk to even family members that aren't even in science, but figuring out how to communicate your work and your record of accomplishments to somebody who has no clue about what you do is really important. That's a great way to practice how you communicate your work, and then just be prepared. And I think, as long as you're excited about that role, you look into it, it'll come through during the interview process as you meet people. But just really take the time to think about what you did.
Yeah. Andy, what would you say?
Yeah, I only have a few things to add. I think, going back to what Ray started, I think the reason to go into industry is that you really do want to make a significant impact in human health. And that's because Shiva notes, we fail 90% of the time. So if you're not really interested in making a drug, you're going to lead a very, very depressive career. Okay? And there are phenomenal opportunities in academia, to really pursue great science. And then in terms of the types of skill sets or personalities, I think in both cases, you need to be innovative. You need to be committed. You need to be rigorous. You need to be you need to work really hard. And I think the other aspects, the two additional aspects that I think are absolutely required in industry is your ability to work on a team, and it's no longer about you. It's about the project and what we can do about the project. So I think that last part is something that I would say, having recruited a number of really outstanding senior academics, 50% fail on that one.
Mm-hmm. Yeah, interesting.
Just to amplify one more point, I think that you might have been getting at, is if I was a student or postdoc out in the audience, don't worry about do I know how to do a high throughput screen? Do I know how to do a flipper assay or whatever other assays commonly used in industry to study small molecule protein interactions? You don't need to worry about that. You'll learn that stuff in industry. I mean, what we're looking for, and I think both Shiva and Andy spoke to this, is we're looking for people who are well-educated life scientists or chemists or physicists who are passionate, who are smart, who are agile. They can learn new things fast, communicate well. That's part of working in teams, and are very good at working in teams and collaborating with others. The detailed stuff, you'll learn it. We'll teach you that.
I'll just take one question from the audience because it got 5 hits. It was called, how has the culture of industry changed in the last 10 years? And it gets very specific here, especially in terms of parental leave and career support for mothers who have to take time off to support their newborns.
Maybe I can start. I think over the past 20 years I've been in Genentech. At least Genentech has learned from the evolving culture and the social aspects of our industry. Sometimes maybe a couple years late. But we have put significant resources into ensuring things such as very important to us as mentorship, guidance, support, work-life balance, and just much more recently, I'd say that over the last 4 years, a much greater emphasis on diversity, equity, and inclusion.
Shiva, do you want to comment to that?
Yeah, I would echo that. I mean, I raised my kids while I was at Genentech for 15 years, so I have 2 boys. One is in college, one is 17 now. And I think that there was tremendous amount of support. So industry has definitely evolved from the time I started. When I had my first child, I was at a small company down in San Diego. It definitely wasn't that at all. But what I experienced was a really supportive environment, but not only that, a focus on helping to develop senior women leaders at the organization. And I can tell you that all my peers at Genentech who are there, who've left, are all just doing phenomenal things, either in industry, outside of industry, in the venture world. And I think it's a testament to the culture there. And the same thing at NIBR is what I see as an incredibly supportive environment, and it has changed. This was not always the case. I think there's a recognition that having greater diversity actually is better for the company. Not only is it good for society and it's the right thing to do, but it actually produces better results. That's where you get better innovation. So there's data behind this, and I think because of that, there's a lot of acknowledgement of the importance of not just supporting women, but diversity as a whole. And I think it goes beyond parental leave. It's really about dealing with many other aspects that hold various, be it gender, based on gender, ethnicity, or race behind. It could be bias, right? There's a lot of training that has been implemented now. There's many other things that we're working on beyond just factors like family leave. Which I think is really important, and it's great. It's taking a far more holistic approach at the problem and not just one aspect of it.
Ray, you want to?
Jim, one thing I think that's interesting is that perhaps in some unexpected ways, the pandemic is having, I think, a really positive impact on folks, especially young parents with children, because we're moving away from this model where you physically have to be in a certain place from 9 a.m. to 5 p.m., every day at minimum, and recognizing that, people can work in a more flexible way. And obviously, if you're an experimentalist, part of that is going to be at the bench. But there's a lot of time where you're interpreting data, you're analyzing data, you're planning experiments. And, the pandemic has taught us that we can be flexible in terms of thinking when people do that and where they do that. And, I think we've seen at Amgen, because we've taken a very open-minded approach on remote work, that's actually been a real advantage for recruiting, and especially for people who, young folks who have families where, it frees up key windows of time in the day where, you can help your children, get off to school or come back from school. You can trade off with your spouse, and you don't have that regimented schedule of having to be somewhere at this exact block of time, but still get your work done in a very effective way.
Yeah, that's great. Okay, I think I went over time there. The panel was so engaging. So let's thank Ray and Shiva and Andy. And just before we break up, I want to make a couple of announcements. Next, there's a poster session that started. There'll be 2 poster sessions at the meeting. please attend. I think there's more than 50 posters over there, and will be followed by a reception. There are going to be 3 poster prizes given for the top 3 posters. Looking forward to all that. And then finally, you know, this thing, we've been at, I don't know about you, but I feel like, as Bill Degrado said, I feel like I've been drinking from a fire hose today. And so looking forward to a little bit of loosening up. And we're going to have some opportunities for that. We're going to have Robert Stroud and the Jailbirds are going to be performing their rock music out in front of Genentech Hall, starting around 5:15 or so, 5:30. And then most importantly, or as importantly, we're going to have the first human bubble Olympiad. What is that? You've probably seen these bubble suits that people can wear, and then they have various games like Capture the Flag or Bubble Soccer or Protect the President. But here we have the opportunity with Stanford, Berkeley. Now, they always compete, but UCSF never gets in there. So they're going to have the Bubble Olympiad out in the quad area. These are chemical biology students. Each team, I'm told, has at least 10 to 15 players, 5 at a time. And so it's 5-on-5. It'll be a round table. It'll be a round robin kind of tournament in 3 different types of games. So be sure and enjoy both those things as you have your libation and reception and take in the posters as well. Okay, thank you so much.